The Nipah virus. At first, scientists thought it had something to do with pig farms. But if that's the case, why is it prevalent in Bangladesh and some parts of India where pig farming is not common? And how scientists figured it out? The story began in 1998 and still going on. If you are not familiar with this virus, then you don't know that this deadly pathogen can cause the next pandemic. It's a ticking time bomb. If we don't take the necessary steps at the right time, it will shake the world.
The World Health Organization shows more than 600 human infections reported between 1998 and 2015.
Nipah virus.
Nipah virus.
RNA virus ആണ് Nipah.
Nipah virus.
No vaccines to prevent or cure the infection, which has a mortality rate of up to 75%.
During September 1998 to May 1999, in Malaysia and Singapore, some patients were coming to the hospital with fevers, headaches, and encephalitis. Encephalitis means inflammation in the brain, which is usually caused by infection. First cases were reported from Nipah River village in Malaysia. Around 300 cases were recorded at the time, and 100 of them died. Initially, based on some antibody screening, doctors and scientists thought the disease was Japanese bee encephalitis. Moreover, Japanese bee encephalitis is a mosquito-borne disease that can affect animals including birds, horse, swine, and humans. It was a common swine-related disease in Malaysia, but anti-mosquito fogger machines and aerosols did not control the situation. More importantly, they found out later that the signs and symptoms don't really fit with Japanese bee encephalitis. For example, at first they saw the people who got sick had direct contact with pigs, which is not the case with a disease spread by mosquitoes. Second, the symptomatic cases among members of the same household were very high, reaching up to 33%. That's even higher than the attack rate of Japanese bee encephalitis virus, which only causes symptoms in 1 out of every 300 infected patients. Third, some of these patients were vaccinated for Japanese bee encephalitis. They had enough antibodies, still they were infected by the virus. Fourth, there were reports of infected pigs getting a severe barking cough and dying from the disease, which was not characteristics of Japanese encephalitis. 5. There were some Malay villages that were in close proximity to these pig farms. However, these Malay villages did not show any reports of this mosquito-borne disease. Point to be noted, Malays are predominantly Muslim and pigs are forbidden for them. In March 1999, similar reports were also coming from slaughterhouses of Singapore. So millions of pigs were killed and deeply buried. The Malaysian authorities, with the help of the army, also killed 1.1 million pigs. The farms were decontaminated and it worked. The outbreak was under control. After 1 year, similar reports were coming from a place called Siliguri, which is located in West Bengal, India. That is around 4,000 km away from Nipah River village. It's a different country, different culture, and different geography. The patients had similar types of fever, headache, and encephalitis. Around 68% of the patients died. This time, the fatality was 35% higher than the previous reports of Malaysia and Singapore. At the same time, the neighboring country Bangladesh was also reporting the same disease. A district called Meherpur, which is around 460 km away from Siliguri, reported 13 cases with 69% fatality. In the meantime, some scientists from University of Malaya isolated the virus from the 1998 outbreak, and obviously the shape of the virus was not resembling Japanese encephalitis. Rather, it was similar to a virus called Hendra. Hence, researchers collected some Hendra virus antibody and performed an antigen-antibody reaction with the new virus. In addition, this virus has only a 20% genomic difference with Hendra virus. At first, they called it Hendra-like virus. Then they named it Nipah virus because the first case was reported from Nipah River village of Malaysia. Remember, when you get a close relationship between a novel virus and a well-studied virus, it becomes a lot easier to study about the new virus. For example, when COVID-19 took place around 2020, scientists figured out that the new virus was closely related with SARS coronavirus from 2002. This made life easier for scientists and doctors to take necessary actions. Similarly, when scientists discovered that this new virus is closely related to Khejurer virus and their illness patterns are nearly identical, they began to compare the two pathogens more closely. For example, Khejurer virus was discovered in 1994 from specimens collected during an outbreak of respiratory and neurological disease in horse and humans in Khejurer, a suburb of Brisbane, Australia. Khejurer virus usually resides in fruit bats. The transmission takes place from bat to animal. So logically, if Khejurer virus can live and get spread from bats, the Nipah virus should do the same. So scientists searched for Nipah virus in bat. And guess what? They found it. Later studies showed that fruit bats from India and Bangladesh also contain this virus. Therefore, the cases from Bangladesh and India were coming from bats. The cases from Malaysia and Singapore also came from bats. Bats are the primary source of this virus. From there, they can transmit via domestic animals such as horse, goats, sheep, cats, and dogs. However, the virus is probably more contagious in pigs than other domestic animals. That's why the Malaysian pig farms face that outbreak. The Nipah virus is still a serious threat in South Asia, especially during winter. Here in Bangladesh and India, Bengali people enjoy a traditional drink called Khejurer Rosh or date palm sap. It is a juice collected from the sap of date palms. This sap is higher in sugar, which is a type of energy source that both humans and animals crave. That's why bats are particularly attracted to the date palm sap. At night, bats are often drawn to the collection containers placed at the top of the date trees. They leak the collection tube of the sap to get the sugary liquid. The problem is flying foxes like this can carry the Nipah virus. If a bat is infected, the virus can be present in its saliva. When these bats leak the sap, their saliva can contaminate the entire collection, making the sap unsafe for human consumption. Nipah virus can stay viable in a sugary environment for a long time. When a human consumes that contaminated date juice, virus enters through the nose or mouth. From our experiences of COVID, we all know viruses enter into the host cell using a receptor. If you remember, COVID-19 coronavirus uses H2 receptor to enter into the host cell. Similarly, after the entry, Nipah virus G protein uses ephrin B2 and ephrin B3 for entry. These receptors are highly abundant in our lungs, stomach, colon, and brain. Remember, ephrin B2 and ephrin B3 are also expressed in animals such as cats, dogs, and pigs. That's why these animals also get sick when the virus attacks. Now, after the binding of G protein, A protein helps to fuse the virus into the host cell membrane. In this situation, the infected host cells release signals using chemicals interferons that call the white blood cells. The host immune system elicits a robust immune response, which results in symptoms like fever, headaches, and encephalitis. Sometimes the host wins with a huge damage to the kidney, brain, and other vital organs. Sometimes the virus wins and we pay the price with our life.
Bangladeshis, especially those who are illiterate, remain hard to reach.
Bangladesh is facing this virus every year. India also facing the same. For example, last year, the state government of Kerala reported 6 laboratory-confirmed Nipah virus cases. In 2024, we already have seen 2 confirmed cases of Nipah virus in Bangladesh. Both of them had a history of consuming fresh, raw date palm sap. There are some easy steps to prevent Nipah virus spreading. The CDC guidelines say proper hand washing, carefully handle sick pigs and bats, avoid areas where bats roost. Do not eat or drink products that could be contaminated by bats, such as raw dead palm sap, raw fruits, or fruit that is found on the ground. Do not contact the blood or body fluids of any person known to be infected with Nipah virus. However, in Bangladesh, raw date palm sap is very popular drink. Boiling raw sap makes it safer, but people do not like it because boiling alters the taste. Juice collectors use bamboo skirts or nets to protect the jar where the dead juice accumulates throughout the night. It protects the juice from bat saliva, but sometimes the bat clings to a palm tree as it eats sap just above a collection jar. While sucking the sap, a bat might pee into a sap collection jar. And like bat saliva, bat urine can also contain Nipah virus. So this protection method needs to be improved in future. There are currently no drugs or vaccines specific for Nipah virus infections, although WHO has identified Nipah virus as a priority disease for the WHO Research and Development Blueprint. Intensive supportive care is recommended to treat severe respiratory and neurological complications. What if in the future this virus gets mutated in such a way that it becomes more contagious and gets spreaded in the whole world and starts a new pandemic? It will be a huge threat to the global public health since both India and Bangladesh have a very high population density. We need more prevention measures and proper drugs or vaccines to stop the further outbreak. Thanks for watching.